physiology of it, just a quick review of these functions of the nucleic acids as our DNA and RNA and so on. How it applies to these steps of making copies of the DNA into replication, transcribing DNA and RNA sequence, and then translating from the language of nucleic acids to the language of amino acids and proteins, how you go from RNA sequences to protein sequences. Okay, so DNA is genetic material, of course. The whole the whole thing inside the nucleus, you would refer to that total genetic information of all the chromosomes together as the genome. So there's a human genome. It took them about 10 years to sequence it. First time in the 90s. Now it can be done in a matter of a few hours. Automated machines. One technician running it instead of thousands of scientists doing it. And soon you'll go to your doctor's office and get a genome sequence, and it'll tell you that you're prone to the following 10 diseases. And this medicine will be better than that medicine. That's already happening in cancer treatment. But they'll sequence the genome, they'll find some little mutation there, and they'll say, oh, it's because of that this cancer started. But we know with that version of it that this chemotherapy agent doesn't work so well, and that one works better. So that's the genome. The individual segments, the teeny, by comparison, tiny segments of the molecule that can be translated into a particular protein, that little chunk, which would be a few thousand bases, maybe only, instead of millions and millions of them. That small chunk that codes for a protein is referred to as a gene. So again, replication is a process of copying the information in one strand of DNA into a newly synthesized strand. And both strands are copied. Everything is copied from one end of the molecule to the other. You don't skip over any part of it. It's done with the enzyme DNA polymerase. High fidelity enzyme, it doesn't make mistakes. What's that, 1 in every 100 million accidentally get a mistake. Even that, there's enzymes that come along afterwards and try and clean up the mess from that. Mm-hmm. So it's even better than that. You know? So you use a template strand to build a new strand. It's semi-conservative in nature, meaning that if you look at the daughter strand that results the new double helix, let's say that's the parental chromosome, you're going to make a copy of it. The copies, the two that you'll see in the next generation, will be hybrids. So this was the exact original molecule, untouched, and here's its newly synthesized partner, making it a new double helix. So if you were to look at one of your chromosomes, you could trace that back and say, okay, this one came from, one parent, and this one was newly synthesized. But one of them was the actual material itself from the parent. Okay, then the other kind of process you can have is just transcribe the DNA. So replication is DNA to DNA. Transcription is DNA to RNA. So it's done with an enzyme called RNA polymerase. So it uses the ribonucleotides, not the deoxyribonucleotides. And it's selective. So this is done when you want to make a protein. This is done when you want to make a new copy of the chromosome. If you want to just make an extra copy of hexokinase, you're going to have to find a gene for that. You're going to do a little local unwinding. You won't unwind the whole thing. You'll just unwind the region around that gene. And you'll copy the information from just one strand, not from both strands. In replication, both strands get copied. Here, only one of them will be the so-called sense strand, and you will take this information and make a replicate of it, or a complement of it, using RNA. So in this case, the red molecule is RNA, and the DNA is the template strand. It still uses the same Watson-based rules, but it's picking up RNA instead of DNA-based variants.